Okay, hello to all the HubLE audience. My name is Diego Grinman. I'm a postdoctoral associate at Yale University in the US, and I'm here today on behalf of HubLE with Ghazal Vahidi from Montana State University to talk about their paper titled Bone Biomechanical Properties and Tissue Scale Bone Quality in a Genetic Mouse Model of Familial Dysautonomia, which was published in the Osteoporosis International. So, Ghazal, welcome. Uh, and to start with this, could you tell us, a little bit of your summary of the work that you've done and how it fits into, the big picture of your field?
Yeah, sure. First of all, thank you so much for having me here and this great opportunity. Yeah, as you mentioned, my name is Ghazal, and I'm just at the start of my third year as a PhD student in mechanical engineering at MSU. And that paper we published was about familial dysautonomia, which is, I'm gonna from now on call it FD, how we mention it usually in the paper, so it's just easier. Yeah. So FD is a really rare hereditary neurodegenerative disease that actually only affects the Ashkenazi descent of the Jewish population. It's super rare, and it is a multisystematic disease, can have negative impacts on nervous system, gut, kidney, posture, gait, balance, and a lot of other stuff. And unfortunately, FD patients also suffer from bone fragility and bone fractures, like up to 60% of them experience bone fragility and fractures during their lifetime. So, and there is no treatment or management plan out there for the bone fragility that this vulnerable population is experiencing. Right. At MSU, we have this mouse colony. It is like a genetically mutated mouse colony of FD mouse that actually mimics a lot of phenotypes that we see in human FD, like the altered posture and gait and kidney and gut dysfunction. So we were really interested to see if this mouse model also mimics the bone fragility that is seen in human FD. And the motivation we had here was because if this mouse model also mimics the bone fragility, that means it can be a really strong platform to do a lot of investigations regarding bone fragility and also hopefully some therapeutics someday. Because, because it's a rare disease, it's really hard to get human samples. So we really need other models to study FD. Absolutely. So that was why we did this project, and what we found was really interesting. We found out that our mouse model of FD is actually mimicking the bone fragility that we are seeing in human FD. So our FD mice had lower bone toughness and fragile bones. And so the question was, the answered question in the field is why their bones are fragile. Bone fragility is kind of about bone mass, but also bone quality. So that's why we wanted to invest. There's also some information out there that the bone mass is also decreasing in FD. Right. So we're really interested to know what about bone quality? Like what happens to the bone tissue scale properties, the bone quality, bone structure. And what we found out from this study was actually bone quality in multi-level scale is decreasing in FD in our mouse model. So this mouse had lower bone toughness, had altered bone microstructure. The mineralization and tissue maturity was not different, but the collagen structure was also impaired, which was interesting. So all in all, what we found out was this mouse model is like a great platform that you can continue to investigate bone fragility, its potential therapeutics, and hopefully someday they can come up with a management plan for this really, really vulnerable population.
Right. Well, this is great because actually, I was trying to look if there's any other models of familial dysautonomia around there. Are there other models as well that can reproduce the same things that you found?
There are other mouse models out there, but unfortunately, because FD is such a multi-systematic disease and it messes up with everything, it's really hard to like have a model that exactly mimics the human phenotype. But I think the mouse model we have at MSU is actually doing a really great job mimicking most of the phenotypes we are seeing. And it's actually like a lot of like this mouse model is being published for a while now, like the informations are out there. So I think it's really good to draw attention to it and just like being able to like other labs around the world to use it and probably hopefully develop some more investigation about that.
I agree. If you can reproduce this aspect of bone loss in a mouse model, then you could be able eventually to find the target, the mechanism, or even, why not the therapy, right?
Exactly.
So, yeah, one of the things that I wanted to ask you is, I mean, what is the most important thing you learned from this study? I mean, you personally.
That's a really good question. This was a challenging study because, as we were talking about, being like having this multi-systematic disease that at the same time incur a lot of factors that all affect bone quality, like calorie intake that affects bone quality and bone health, and the posture and imbalance all affect. So it's kind of like challenging to like pinpoint what separate contribution each of these factors have to bone fragility. So it was a challenging study, but I learned a lot from it. It was my first project, first time doing some bone research. So I learned a lot about like building my skill set around multi-scale bone characterization, about how to do a study design, how to do a literature review. And I think it plays a really important role in my career as a bone scientist, because it kind of demonstrates my skill set in doing this multi-scale characterization techniques. And yeah, I learned a lot about bones while I was doing this project.
You became a bonehead, That's something very particular about this community.
Exactly.
Well, as you were talking, that most of the times we struggle with projects in a way or another. I mean, the results cannot be reproduced, and then we try different approaches. For you in particular, what were the greatest challenges you encountered with this work, and how did you actually overcome them? Because, how does it work?
Sure. So yeah, one of the challenges, as I just mentioned, was like FD being a multi-systematic disease. So I wasn't able to like conclusively say bone fragility happens because of like disturbed gut, or bone fragility happens exactly because less calorie intake. So I learned that like, first of all, like this is how it goes in when talking about body biology, and you should be able to see a little bit bigger, think outside of the box and try to consider everything there. One of the like tangible challenges that we had with this mouse model was this mouse are really weak and their skeleton was really fragile. So it was kind of like hard to do the normal bone handling process or tissue harvest that we are doing. So we had to be extra careful when doing the bone tissue harvest, when doing different mechanical testing on this super fragile and weak bones. And so like I like learned about like having this extreme fragility phenotype. I learned a lot about how to be extra careful and put extra care when I'm handling these bones.
Okay, so that's really great. I mean, again, to overcome those difficulties is always a challenge, and you're a third year, so this is the best time to face those problems now. And talking about problems, and this is actually sometimes a difficulty. And just very quickly, what was your experience in publishing in Osteoporosis International? Thumbs up, thumbs down.
Actually, it was a great experience because the feedbacks we got from the reviewers were like thoughtful, were constructive, were in depth, and like our paper coming out of the review was in a place we really were satisfied with. So it was a really positive experience.
Awesome. Well, so I guess, I mean, you have good expectations that people actually going to use this model in the future to dissect the mechanism of bone loss in FD patients, right?
Yeah, Yeah, for sure. I think this paper is the very beginning to understand that this multi-systematic impairment to the bone quality is happening. And I think there's still a lot to do. And I think future work can benefit from using mouse from different age spans,
Right.
or like the fresh tissue, or like other characterization techniques. Hopefully people can go and study these bones, study their bone cells, bone remodeling, and they can like get closer to the answers out there.
All right, right. Well, and finally, Ghazal, I mean, in terms of your career and future goals, this is a very tricky question sometimes. I mean, how will this publication in particular help you to achieve those aims? I mean, how is going to help you to further, your career?
Yeah, it's a great question. So the main interest of my PhD research right now is actually studying the bone fragility that comes with aging. And if osteocytes, which are this master orchestrators of the bone remodeling, this permanent resident of bones, if they play a role in bone fragility. So this project helped me a lot to build my skill set about bone fragility and multi-scale characterization. I was able to study bone fragility in an extreme phenotype. So I learned a lot from that, and I can like apply those skill set to my current project, my PhD
Right.
project, which is about the bone fragility and aging. So basically, like my overarching career goal is to lead my own lab someday on my own research group that is dedicated to study bone fragility, their osteocyte roles, potential therapeutics. So I feel like having this paper out there demonstrating my skill set about like multi-scale characterization, it's actually a really, really good outcome of the project for me.
Yeah, I agree. Well, that's great. That's great. And thank you so much for taking the time to discuss your work today with us and with me in particular. And we hope to hear more from you and from your group. Actually, I was going to ask you, do you have a Twitter account that you want to share with the HubLE audience?
Yeah, sure. It's first of all, it was such an honor for me. Thank you so much for this opportunity, and hopefully we'll see you more from the publications from our lab,
Yeah.
which is Dr. Herbrand's Biomechanics and Bioinspired Materials Lab at MSU. And my Twitter account is basically my first name and my last name. And if I want to give the actually exact ID, is like at QAZAALVAHIDI.
That's perfect. Well, thank you so much again.
Yeah.
And we would like to thank all the audience for taking the time to join us today. And if you'd like to be involved in HubLE publications, just like as Ghazal just did, please feel free to contact us. Also, keep an eye on the HubLE team at the musculoskeletal conferences in your area. Find our editors, and we will really very happy to interview you all there. So follow HubLE in the LinkedIn, Twitter, YouTube. Thank you, Ghazal, again, and hope you see you around.
Thank you.